Again, welcome everyone. Thank you for coming to our event of Synthetic Biology Society. Today we are hosting Recep Hoca or Recep Erdem Ahan. Yes, many people know him as Recep Hoca. Recep Hoca graduated from Istanbul Technical University in 2015, double majoring in biology and chemistry. And then he came to Bilkent for his masters. He worked and still working with Urartu Hoca in UNAM. He completed his masters and his PhD as well here in 2023. Right now, he is doing his postdoctoral research in the same lab. Also, he was a visiting scholar at the University of Chicago previous year. And he has a lot of publications up to date for his current academic career. He's kind of famous in the field of synthetic biology already. And today, also, I need to mention one thing. He also participated in IGEM and mentored a lot of IGEM students, as well as me and my other friends. And he was also one of the reasons from the IGEM later on to establish this Synthetic Biology Society. Now we will listen to his presentation about designing bacterial living therapeutics for cancer to treat breast cancer.
Yeah.
Thank you for coming.
A nice inspiring talk for you. It's like you probably know the last paper that we published from our lab. That one is published in this breast cancer stuff. Actually, it's a story that comes along with us. It's the 8 years. Like we just built the first one, built the second part, then another master's graduated, and lastly we were able to publish this final work. So some African people try to stand me. Anyway, as Sam mentioned, I'm currently in this synthetic biosystems lab. Probably you know that the Urartu Hoca is the PI of this lab. I have been here over than 9 years. Actually, my plan is to stay here 3 years, then go to the PhD in US. But I was unlucky or unsuccessful, I don't know. I couldn't get any position from there, and then I started to do my PhD in the same lab. But I got accepted from the other Europe universities, but especially I want to do my PhD in US, that's why I just US, either I will go to the US or I will stay and I stayed here. In meanwhile, I married and my life is like turning a separate path completely, which is like good way of thinking. Anyway, today I will explain you this concept, which is designing bacterial living therapeutics for cancer. Actually, this concept is quite old because you know that if you think something, if you couldn't Google it, find in Google, you need to try harder. Because every idea that you have probably told somebody else in near or far past. For this concept, by the way, you can still do this, like improvement, make a completely new approach than idea. But most of the time, generally, the idea itself wouldn't be new. There's like quite example of this in the science. And if you like saw a paper and told us, oh cool, people like think very innovative ideas to do something. But most of the time, this stuff probably published 20 years ago or 100 years ago had been taught by somebody else. And this concept is not an exception. The first bacterial cancer actually have been tried in 19th century by this guy. Actually, he's a doctor from US. He tried to inject Staphylococcus bacteria to the neck cancer patients, and some of the patients of his actually get cured. They don't understand why they are cured, but they just don't have the tumor anymore after they inject this bacteria. There's a like good explanation now. People understand why it is the case, because these bacteria that are injecting, basically activating the immune system to attack to this solid mass in their neck, and they kind of use this bacteria as an adjunct to activate the immune system. But they are very naive in their case because they are just using a pathogenic wild-type bacteria. It's most of the case, his patients dies because of the infection rather than the cancer itself. But the concept is very nice, and you might thought that it's a very nice foundation to build upon. Actually, in here, if you take this concept and apply a synthetic biology approach, you can design a living, smart biotherapeutics to basically target cancer cells, then kill them specifically without harming the other parts of the body. In that sense, you need to engineer a sensing, also targeting module. Here we will sense the location, also the characteristics of the cancer through specific inputs. These can be either a protein that is expressed on the cancer cell, or it can be a pH change in the tumor nucleus, tumor necrosis region, or it can be a, for example, temperature or a specific metabolite. In that sense, you can sense, you can engineer this part by synthetic biology to sense presence and location of the cancer, basically. And after that, you can add a computational module to make sense of this signal to able to produce a response, which can be building a genetic logic case or memory base to sense, locate, then produce a response. And in the response module, you can basically either convert a non-toxic molecule to a toxic molecule in a spatiotemporal manner, or you can basically secrete a protein, and in case of an unwanted effect, you can also put a biocontainment switch, which can basically kill all the engineered organisms in the body. Okay? It's like the concept itself built upon this, and there's a lots of different groups in the world that are trying to realize this concept. That is first proposed by this William Collin, 19th century. It's a very nice field to watch. There is at least I believe we got 3 to 4 papers per year in each year for at least 10 years, and they are improving all this sensing, computation, and response module to get a nice cancer curing. Because most of the time the cancers that probably you get this lecture and knows better than me is a complex disease, and one of the parts that can be targeted is the immunological part of the cancer, because cancer can grow because your immune system basically cannot produce a cytotoxic effect on the cancer cells. If it's recognized and kill the cancer cell, you probably won't develop any tumor. In that sense, we are trying to build this with a probiotic strain. I don't know, do you know that? It's a probiotic strain of E. coli called Mutaflor. It is available in Germany plus Australia, but I said you can find in Turkey as well. They try to sell it. It's a nice E. coli. It basically have isolated from in World War I from soldiers that not developed diarrhea. It has inherent agonistic effect against gut pathogens. Therefore, it's been in human use over 100 basically years without any serious adverse effect. Actually, prior to the antibiotic revolution, you know that they able to produce antibiotics in scale and distribute it to kill infections. There's other concepts that scientists try to cure infection, and this idea is basically one of them. They thought that there's a special organic microorganism which can kill pathogens, which can be used to cure other infections. It's another idea. Because the antibiotics are so successful, this kind of research are basically buried under the dust. But there's another approach, which is the use of bacteriophages, also getting popularized because of this antibiotic crisis. Will be faced, hopefully not faced in the future. Okay, this one is called Nissel because this guy called himself Nissel. His name is Alfred Nissel. This bacteria is important because it has an inherent capabilities which make these organisms very ample to engineer as a living therapeutic cases, as well as because of these inherent capabilities, it can produce a favorable effect when you like consume it or use it as a therapy. These can be, these are like, it has an antagonistic effect against gut pathogens, like secretes myosins. It has a lack of any pathogenic ID, which is one of the important attributes of an antibiotic. It lacks of O antigen. Do you know what is O antigen? For curious, why you don't know that?
Yeah, I guess it's the
Do
one.
you know what is septic shock?
Yes.
Because
Why do you get septic shock?
Because the immune system might be overactivated. So
Of what?
of the antigen.
Which antigen?
L6, I guess.
Yeah, it's like lipopolysaccharide. If you have like blood infection, these nasty gram bacteria has a like endotoxins of lipopolysaccharides, which basically recognized by your immune system through TLRs, like receptors, which basically produce non-specific heavy immune response, which basically kill the That's why it is like important attributes of a probiotic bacteria. It has a sequence genome, which is the most important, by the way, attributes of a probiotic organism if you want to engineer. Because without the genome sequence, you don't know what to engineer. And lastly, it cannot produce any serum infection, and it has a two cryptic plasmid, which is like nice to get.
So do these cells not entirely produce LPS at all, or they just like produce a variant of it that's not recognized?
The second one, they produce an lipopolysaccharide, but it is like not produced as much as high as other LPS. They not produce immune response, not as high as other LPS due to this mutation, basically. Okay. Okay. In the like first part of the story, it is displaying a display shuttle because we want to like locate where is the cancer cell, through which can be done either sensing a specific signal or recognizing a surface antigen, which can be a protein or a polysaccharide, okay, or the protein saccharide complex. In the first part of the studies, we have developed a display shuttle. Display shuttle meaning that you are basically producing a protein which is expressed on the surface of the cells, like receptors, okay? But in that sense, you can put a desired proteins on the cell surface of the bacteria, which later can be used to locate the cancer cells, okay? To develop an display shuttle, we engineered an autotransporter protein. Autotransporter proteins are the proteins that are expressed on the cell surface of the bacteria. By themselves, they have inherently chaperone activity to secrete themselves to the extracellular matrix, okay? Most of the time, if a bacteria do this kind of stuff, they do it because of the pathogenicity advantages, okay? If you find a protein that is overly compact plus on the extracellular media, it probably has a link with a pathogenic activity. In that case, this protein is expressed when the environmental conditions are not favorable for the bacteria, and bacteria express these proteins to the basically compact each other and produce a biofilm. But luckily for us, it is a well-characterized protein. It is a L-shaped outer membrane, extracellular domain, which is like this pathogenic activities encoded in this first 160 amino acids. To display a shuttle from it, we basically delete that part. We put a carboprotein. Here it was a SHGP, which is a resonance protein. Then we express it with a single peptide. You can think that this part of the protein is gone. This part, this little curly stuff, and we put an SHGP on it. Okay. When we express it, we saw that very nice, clear GFP signal from the cells. When we labeled with an antibody, we saw that cells are labeled with this antibody, which is showing that our proteins on the cell surface. Okay? Is everybody cool? Cool. Then in the next part, we have like published this in 2018. Then start the work on this. Okay, this part is the basic part probably you are have seen in the late publication. Here we change this SFGFP with a nanobody. You know what's nanobody? It's a like, yeah.
It's essentially an antibody that has been stripped of all parts except the part that
Where they get this nanobody?
Sorry?
Where they get a nanobody from? You know that? Do you know the story? It's a very cool story, by the way. They get it from the camels. Do you know how they found it?
I didn't. I'm not really sure, but they found out that the antibody structure is different in camels than humans. So they started to use that.
Yes, it's correct. There's a guy in Belgium basically doing some ridiculous camel stuff which nobody basically interested in. And he noticed that, oh, camel has an antibody which has a single domain antigen recognition. In human, antigen recognition domain is what? Of an antibody is what? Variable is big, right? It's composed of two domains, not one domain. It's a light chain and heavy chain. But in camel, there is just one domain, which is encoded these all variable regions. And they sell this technology to the Abbey for over $16 billion. It's a quite interesting study. Nobody carries something in camels, and then somebody gets rich. Rich. Not just rich. Yeah. And this was like, I was in undergrad when I saw this, one of my first papers on this, and I was just like, oh cool, they found something interesting. This is like very nice attributes to be used. Actually, there's like the FDA approved drugs based on this nanobodies. Okay, they are like coming from camel. Yes.
Do they induce immune system in the humans?
Nanobodies. Yes, probably because they are not accepted as a themselves. They are foreign proteins.
Then you cannot use bacteria expressing nanobodies.
It's completely different story. Okay, if you like put a nanobody, at some point it might produce an immunogenic response because everything that is not self can produce an immunogenic response, but not necessarily. So I have told this story to you because you know how CRISPR is found. CRISPR-Cas9. Do you know this Fanzang story?
Yes.
You know that? It's very cool, by the way. It's also like a boring bacterial immune system symposium, which is like, I believe there is 4 other people in that room plus Fanzang. But at the end, he was able to re-engineer this protein to be able to basically revolutionize this genome editing stuff. Which is also another cool story. That's why when you attend a seminar, you might think it's very boring. It might be a very boring, by the way. Most of the time, these seminars are boring. I was like attending a conference like a month ago. It's far from you, okay. Talks are not so like entertaining, but you try your best to listen them, okay? Because you don't know when an idea stuck your head to like might change your life forever. Okay, it's a nice story. Okay, we like where were we? We put this nanobody on the cell surface, and we show that it's expressed. The cool part is that we have used this HER2 nanobody. Do you know what's HER2? It's like a protein that is expressed on cancer cells. Okay, it's a very famous antigen because it is overexpressed in cancer cells. There's like an approved monoclonal antibody targeting these HER2 proteins to basically cure the cancer cells.
Was it breast cancer?
Yeah, it was breast cancer. There's another lady basically died because of this antigen. Do you know CAR-Ts? There is a like, yeah, the chimeric antigen receptors. When they like able to respond from this Amy Whitehead, which is like first person to get a CAR-T cell therapy to his leukemia. He was cured completely in 2012, I guess. In around 2015, people thought that this CAR-T approach will cure the cancer completely, and they changed this antigen recognition domain of CAR-T to the anti-HER2 and infused the cells to the patient which has a breast cancer, and she suffered a very painful death because unfortunately this HER2 stuff also expressed on your lung, and your immune system basically, your engineered immune system basically attack your lung and kill the patient in 2 days. There's a nice clinical paper on that. Okay, it was like our main idea. We don't want to use these CAR-Ts. We want to use a bacterial therapy which has less side effect when things go wrong. Because when your immune system starts attacking you, there's no way you are going to stop it. Okay, it will like slowly or fastly going to eat your. Anyway, we like display this HER2 nanobody on the cell surface of the bacteria, then try with GIT1 cells, which is like HER2 overexpressing cell lines. As you can see, when we like treat cells that is expressing HER2, nanobody, our bacteria are attached to this. Can you see there's a like red stuff in there? No? You can check it from the computer. It's okay. There is like lines of bacteria basically attached to this bacteria. The cancer cell, okay? We basically able to localize and target a cancer cell with this HER2 or cell surface antigen, okay? After that, you want to secrete a specific toxin, non-specific toxin, so that when we like target the cells, we would able to secrete a toxin to kill these cancer cells, okay? In the proximity, we choose a pore-forming toxin because in pore-forming toxin, proximity is important. Because when you secrete low amounts of pore-forming toxins, it produces a nice killing effect because that is attached to this membrane of the cancer cells and forming pores to like kill the cancer cells. In first step, we basically produce this toxin, it's like EDE protoxin, which have been used by pathogenic bacteria to basically burst your red blood cells, okay? So like bacterial toxin, we have recombinant expressing, treat cells with this toxin, we saw a nice clean effect, so the purified toxin. Then we put 2,000 nanogram per microliter, cells basically died. Then we segregate this toxin to the extracellular space with a carrier protein. Then we like treat this secreted proteins with the cells. We also see the similar effect, which is basically showing us toxins are in their active form and able to form a force. Okay. Then we were able to segregate the toxin. In here, it is like basically the proof of the concept that our system is working. We display a targeting module on the cell surface and segregate a toxin. In that sense, when we treat cells with a decreasing number of the cancer cells, what we will expect? If there's a targeting module, the amount of bacteria that is required to produce a cytotoxic effect will decrease, right? Yes. And what you can see here, basically. If you put a nanobody in the lower number of bacteria, it produces more cytotoxic effect because bacteria can localize on cancer cells and secrete a toxin which basically kills the cell. But if you compare to the cells that are not has a localization domain, they cannot kill because they secrete their toxin to the basically spermatant of the cells. Okay, and we basically proved this concept in 3D model. We have used spheroids. In that, spheroids are more realistic model of cancer because it has they has a nucleotic core, necrotic core. In here we also do this like we put demand wild-type E. coli without targeting module and E. coli with targeting toxin. When you compare this array of the spheroids, you can see that when you put a targeting module, your cells can attach to the spheroids, secrete toxins and kill, which basically result in loss of the green fluorescence of the spheroids. Okay, and that's all. It was our study, and we are currently pursuing this idea with other cancer types, and also we are trying to engineer different way of localize and deliver therapeutic agents to this cancer stuff. Also, I'm currently interested in other living modalities to like attack and target and attack cancer cells. Okay. Actually, it was a collaborative study of our lab. I believe there is many people that I was here. Yeah, actually, Gözel, she was the like lead author here. There's Julian Ostoku. It's also these two guys are from Weekend NGO, by the way. Also, like we start this targeting module with Julia, then we continue to Gözel for this secreting module plus this proof of concept studies. Okay, and these all other are the people who have been our lab for at least 5 years. Okay, that's all, guys. Yeah, welcome. Go ahead.
How do you ensure that it only kills the cancer cells?
We have a control group. I will include it here. We first of all, if you like check in here, you cannot target the cells without a cognate surface receptor. Okay, it is the first one. The second one is actually we put and control experiment in here. We have tried this system with a cell line that are not expressing HER2. It was MDA MB231. It's a triple cancer cell line, triple negative cancer cell line, and we didn't see this effect.
But normal cells, don't they express HER2?
They are expressing HER2. It is like a problematic version of the idea. That the good part is that you can engineer the bacteria to produce toxin in this cancer necrotic core. Normally, your tissue has no necrotic core, but cancer has. And necrotic core has its own environmental clues which you can sense. For example, it lacks of oxygen, it has a high acidic, I don't know, maybe high acidic, I'm not sure. And other molecular clues that you can sense through biosensors. For example, it has like intracellular metabolite, which probably you don't have in your normal tissue. Okay. Yeah, I hope so. Yeah, definitely. That's okay.
Does the score differentiate from different types of cancers, or is it mainly the same?
The necrotic core, mainly the same because necrotic core occurs. It's like by the way, it's true for this solid masses. Okay, it's not true for this. For example, circulating cancer cells. The necrotic core occurs because when this like lump gets bigger and bigger, there is not enough veins to support the cancer growth because the veins are not like penetrate to this solid mass. The cells that are inside the mass are basically get necrosis, and they produce this weird environment in the core of the cancer, and which causes the evolution of the cancer as well. Yeah, go ahead.
So, normalized spheroid area.
Yeah.
So it's not approaching, I mean, it's not zero.
Yes.
So it's not like 100%
It's
efficient.
an in vitro assay because some cells are still there.
Because it's,
Yeah. Because some cells still, when you do this measurement in image, you can get a signal. But I believe there is no viable cells in here.
So you just measure the signal.
Yeah. That's why like they have like probably there's a like background signal here coming from measurement. That's why it's not zero because you can.
Why don't you measure it from other ways?
We tried. It's
Not
hard
only-
to work with this first stuff because when you try to like measure the cell viability in these cells, they like basically get disrupted. It's very hard to handle. When you like form them, you cannot transfer them, or you can like change their media. They just basically burst and goes away.
Are they totally fully made from tumor cells,
Yes.
or they also have plastic cells?
Just cancer cells.
So you haven't made any experiments in vivo?
No, there's no.
Maybe mouse?
No.
Is there a way to do it in vivo?
Yes, it
Nice.
would be a nice work for a PhD student if he or he wants to spend the 3 years, 4 years of his life. or her life. I mean, with other software and experience. I hate in vivo stuff. That's why. If you ask me to do an in vivo, I probably search for a cohort of you like willing to spend that much of time to basically do this stuff. Yeah. Yeah, first year student. Tell me.
We are still seeing a decrease in the normalized spheroid area.
Yes.
Even without adding the bacteria.
Yes.
Why is that?
Because when you like form these spheroids in like demand, after like you incubate in this media, the cells that are like trapped within the core dies, which is the reason because of the diffusion effect. Because the cells that are in the like age can easily access nutrients, but when you like approach the core, the nutrients cannot penetrate as well. They die and they are like GAP signal decreases. And also you can see that they like area also decreases. Okay. This kind of in vitro stuff has this weird stuff by the way. They are like completely represented in vivo or like clinical stuff. But it's like nice, cheap, and easy way to show whether your system is working or not. Yes, sir.
No, I would assume that the mutation rate is very high when you put the bacteria. And if you do it in vivo, it will be higher than doing in vitro. How would you make sure that the bacteria doesn't go, I don't know, pathogenic or it's still worse?
To become pathogenic, you need to like attribute at the first place. If you don't have pathogenic tools to begin with, you cannot become a pathogen out of nowhere, okay? The evolution is not working like that. And if you put somewhere without any attributes, you cannot develop these attributes through the like couple of generations or even like hundreds of populations. You need to like find, for example, it might be the case, for example, they're also trying this salmonella cases. You know salmonella? Very nice bacteria. They also like try to use these salmonella strains to produce this kind of effect as well. Actually, there's a like clinical trials with this salmonella. They are injecting in brain tumors because it's a very like pathogenic bacteria. It can produce a nice immune response to kill glioblastoma. It's a very weird way to cure a cancer. Basically, they're injecting a pathogen on your brain, but it works by the way. Because I believe a couple of people have cured glioblastoma from there.
From salmonella, there was a paper I also read
Yeah.
for cancer in
Gut?
gut. Yes.
Yeah. Gut also like a nice place to use this.
I guess those are also
Yeah, it's
working on
working
that for
yeah.
people.
And it might be the case for that because when you like have some sort of pathogenic tools, proteins, which is like silenced through your evolution, can reactivate if you like put a very competitive environment. But if you don't have any pathogenic attributes, you cannot evolve them.
Also for
By the way, the evolution of the living therapeutics are not like most of them are not main concern because they are like cleared when you like put in a tumor because if there is no tumor, your immune system will like hunt these bugs in your body.
also, for as I know, spontaneously cause immunogenic response.
Yeah.
Could it be the case for this also?
For this might be, because even if it has a like low immunogenic response, because it's a bacteria and the cells are dying through necrosis, if the like tumor environment become more immunologically active, the tumors will be shrink quicker with the like help of this bacteria. And also it's true for the oncolytic viruses as well. They also like there's a couple of viruses which can specifically can infect cancer cells which later cause an immune response to basically cause the elimination of the cancer. Yeah.
Hocam, are you are you using antibiotic sensitive or resistant E. coli?
Here,
Yes, here.
they are like, because this is a model, we have used plasmids, which has antibiotic resistance genes.
Mhm.
But if you want to try it in vivo, you probably get rid of these antibiotic resistance genes.
Okay, and can we use antibiotic in case there are some off-target events and
Probably, yes,
off-target
that is
effects?
the reason that people are favoring, it's like one of the reasons that you can favor this over the other therapeutic modalities, okay? Because you can like directly stop the activity of this living therapeutics just using course of antibiotics.
Mhm.
To engineer this kind of like safe switches is very much more higher in CAR Ts. But if you ask me, CAR Ts are better than bacteria because they are
Why?
inherently. Why? Just because they inherently your immune system. Their main job is to basically do a surveillance in your body to detect and kill something. You like re-engineer these cells to do it, but they are evolved to do it. You can like, if something evolved to do a function, you can easily improve that. But if it's not, there is no, you need to build whole system from scratch. But there's no one key that fits all the stuff. Probably in somewhere using this concept, it will be much better than using CAR-T. And also CAR-Ts are expensive.
And for CAR-Ts,
Yeah.
there are not many options, right?
Many options
engineer because there are not that much, there are not enough markers for each specific cancer type, so that you can engineer for it specific CAR-T. Well, even though if they express on certain type of protein
Yeah.
as an antigen, it can be expressed in other healthy cells.
But you can like engineer a computation in CAR-Ts.
Like how many operations can it do? Two,
Yeah, like
three?
it's a mammalian cell. It's not a like Scotch bacteria. You can put larger amounts of large DNA payloads and mammalian cells. You do the computation. And there is not just DNA-based computation. You can also do this computation on the protein level as well.
True, but not all proteins as well, the cancers expressed or secreted.
It should be, there is no must to be a protein. There's lots of different clues that you can sense through CAR-T cells to produce a response. If you ask me, CAR-T cells are much better than bacteria, but they are expensive. Expensiveness is also a problem because you cannot cure everybody with a therapy that costs $500,000. Okay? But it is a lot of
Probably expensiveness because of
there is
competitiveness.
I mean, there must be a solution cheaper to the patients that have no access that kind of thing. It might be a case, but
It can be maybe kind of risky but effective.
Could it potentially lead to, for instance, if you had a She2 site, but the cells without that She2 receptor site would get selected here, right?
You mean that there is HER2 receptors?
HER2, yeah.
Yeah.
HER2.
What happened to the I couldn't understand your question.
If HER2, wouldn't that mean that since cells with HER2 receptors being overexpressed, HER2 receptors genes being overexpressed would get killed?
Yeah.
That would mean that cancer cells without
Yes,
that mutation
it's
would
probably
get.
a problem when it on. These cells might get a resistance to this kind of therapy. But the good way is that you don't just one modality to cure cancer. Most of the time, you are either 3 or more type of anti-cancer therapeutics to cure your cancerous cell line. Most of the time, they will be accompanied with a nice cytotoxic drug to basically kill the actively growing cells. The problem is that, this cancer drugs happen to be act on the targets that are related to the cheap one especially. If these cells are dividing, you can kill them. If they are not dividing, they are in a dormant state, some of the drugs are not acting on them. Okay? It is the problematic because even if you load the body with loss of this drug, your dormant cells still be alive. And if you remove this drug, these cells will divide continuously, which probably make your life very troublesome. Because even if you leave 1 to 3 cancer cells, it will divide in a month to grow a lump again. Okay? You need to kill all the cells in your body to be able to cure the cancer. That's why the immune systems are better option here. If you inject cells that actively do the surveillance and kill the cells when they are become active, would be a better solution. This Amy Whitehead that I mentioned in Cartier, she did not develop any cancer for 13 years. It's a lot of time for a cancer therapy. She's basically cured from cancer. And she was kind of a terminal patient. If there is no CAR T cell therapy have applied to that lady, she would die. Probably she would die. But she's good. She's I will go on up. She was child back then. Yeah, try to read that lady's life. It's a very nice story to get this far. Okay?
Okay.
Anybody else to ask any question?
There are no questions?
No, I can ask it.
Thank you very much.
It's okay.